Let's continue our discussion of muscle contraction. And quite honestly, although there's a lot of stuff on this slide, I want to focus on the right lower corner of the slide. Because what I want to show is what happens to each of the zones we talked about in the last slide when a contraction occurs. And what you can see is that if you measure from Z-line to Z-line, that the distance from Z-line to Z-line is shortened during a contraction. And this is primarily because the distance or the size of the H-band decreases. At the same time, you'll notice that the A-band remains unchanged and the I-band also becomes shorter. So during a contraction, the I-band and the H-band become smaller. This leads to the two Z-lines becoming closer together. However, the A-band does not change. And if you look at the two myofibrils below the sliding filament theory drawing, what you see is that the light stripes become shorter, but the dark stripes remain the same size. Now, I'm going to go through how this happens about 3 times because it is a little bit complicated. However, it is worth understanding how this occurs. Where you want to start on this diagram is with 2 proteins. There's troponin and tropomyosin. Troponin is one of the 2 proteins, and it acts by binding calcium during the contraction. Tropomyosin, in response to the binding of calcium, it's on actin and it rolls back and exposes an active site which is then available for binding by the head of the myosin molecule. So where we want to start here is we want to start on the left side in the middle, and what you see here is that ADP and a phosphate is bound to the head of the myosin molecule. When the active site becomes available, the myosin molecule reaches up and binds to that active site. This is at the top. The ADP and the phosphate is still present. When the ADP and the phosphate are lost, the myosin molecule forcefully moves from right to left, pulling or contracting the actin molecule, pulling it in closer together. As we go down to the bottom, an ATP molecule is then bound to the head, and that binding causes the release of the myosin head from the actin molecule. The ATP is then split, and the myosin molecule reassumes its configuration, and it basically what has occurred is something very much like the rowing motion of a rower. And he has now returned his oar to its original position in the anticipation of yet another pull. So now let's look at another slide of this same thing. And we're going to start at number 1 up here. Number 1, the myosin head attaches to the active site on the actin myofilament and forms this cross bridge. Notice that it's, again, designated a high energy configuration. The ADP and phosphate are bound to the myosin head. Number 2, moving clockwise, shows the inorganic phosphate is lost and is released, and the myosin head pivots and bends as it pulls the actin molecule, sliding it in this diagram to the left. Moving down to the bottom, ATP is then bound to the myosin head, and when the new ATP attaches, the cross-link is lost, and the cross-bridge detaches. And then we move back up to our original position, number 4, and the ATP is split to ADP and phosphate, and the myosin head is then cocked back into its original high energy position. Now there are 2 terms that are used here. The first of these is called the power stroke. And the power stroke occurs at number 2, when the myosin head moves with the loss of ADP and phosphate. And this leads to the shortening of the sarcomere. The other is the return stroke, and the return stroke occurs at number 4, where the ATP is then split, and the recocking of the myosin head in a high energy position, that's the return stroke. Looking at it in another way and using this as somewhat of a review of what we've just been talking about. Now here we're going to start on the right, where it says ATP is coming in and binds to the myosin thick filament. ATP binding releases myosin from actin. Then it moves down to the bottom. The ATP is then hydrolyzed to ADP and phosphate, and this does that return stroke, gives it that high energy position. It moves up to the left. The phosphate is released, and there's an attachment to the actin subunit. And then the ADP is lost, and the power stroke occurs, and this generates the movement. So if you review those last 3 slides a few times over and over again, hopefully you'll be able to visualize exactly what happens during contraction. Now, how is contraction actually initiated? Well, what occurs is the nervous impulse comes down from the nerve, and there is something known as the neuromuscular junction. Now, the neuromuscular junction is the nerve muscle equivalent of a synapse. But instead of 2 neurons coming together, which is what happens with a synapse, a neuron or an axon and a muscle fiber come together. And that space, there's the space there, just like there's a space between two neurons. The axon comes down. Start with the left diagram on the top. You can see the axon coming down and innervating the muscle. If you move over to the top right, you see that the axon terminal with a nerve impulse, and there's something here called a synaptic vesicle, which contains a material known as acetylcholine. Acetylcholine acts as the neurotransmitter. We're going to go over all this again. And then when you move down to the bottom, you see in an enlarged view, this neurotransmitter is released and goes down and binds to receptors on the muscle surface. And this binding leads to an opening of sodium channels. And you remember from our previous lecture that when sodium channels are opened, that this decreases the potential difference across the membrane until you reach a threshold, and then an action potential occurs. So a neurotransmitter, which is the new term and everything that we've just been talking about, is the chemical that's released by a neuron, which diffuses across the synaptic cleft, or the space between the axon terminal and the muscle cell, and it enables the neuron to activate the adjacent muscle cell. And we're going to look at more detail in how this occurs. Now, this diagram is very useful because it goes through steps that happen that causes the nerve to activate the muscle. And we're going to start from number 1 and go to number 8. Number 1, as we know from our previous lectures, an action potential comes down the axon and eventually arrives at the end of the axon. When it arrives at the end of the axon, it opens a specific channel which allows calcium to enter into the end of the axon terminal. This is number 3 in the diagram. The calcium then moves to the little pockets called synaptic vesicles, which hold the neurotransmitter. And these vesicles, in response to the binding of the calcium, move to the end of the synaptic terminal and dock on the end. Once they've docked there, they open up and release the neurotransmitter, which migrates across the space, binding to specific receptors on the muscle, which allows the entry of sodium into the muscle and starts the action potential occurring. Now that's all fine and good, but how does the process stop? In other words, once the neurotransmitter binds to the muscle, how do you get the neurotransmitter off the muscle? Well, this diagram shows you how you do that. For this case, the neurotransmitter is called acetylcholine, and it's one of the more important neurotransmitters that the body uses. Now, acetylcholine is broken down by an enzyme called acetylcholinesterase. And if you look on this diagram, you remember from the previous diagram all the parts that happen. So now in the light white lettering is what occurs to stop the signal from continuing. So once the acetylcholine binds to the postsynaptic membrane, it is then subsequently acted on by acetylcholinesterase and broken apart to acetate and choline. The choline is then returned to the end of the nerve fiber, and the nerve fiber takes it in to make more acetylcholine. Once this acetylcholine is broken apart, the channel that was allowing sodium into the muscle cell closes, and that action potential stops, and it all returns to its resting state. So this is how this all stops. Now, an interesting thing here is VX gas, which is the famous poison that was in the movie The Rock. It was also a poison that was recently used to kill the North Korean's half-brother, is an acetylcholinesterase inhibitor. And the way it works is it causes the body to continually spasm because it cannot stop the conduction of action potentials by the muscle. And that's how it kills a person. Now, muscle relaxation is actually a very simple process. You notice above, at the top, remember when acetylcholine is broken down by acetylcholinesterase, then sodium stops coming into the cell. In response to that, the calcium that was outside the cell that was activating the thick and thin filaments is reabsorbed into the sarcolemma or sarcoplasmic reticulum. When calcium is present, the active sites are exposed. When calcium is not present, the active sites are covered, and therefore cross bridges cannot form. And so when the sarcoplasmic reticulum reabsorbs calcium, there's no active sites available, and all the cross bridges are broken. The sarcomeres lengthen, and the muscle naturally relaxes. And this is how relaxation occurs. Now let's talk about something called a motor unit. A motor unit is defined as one motor neuron, the axon from one motor neuron, and all the muscle fibers that it innervates. And that's shown in this diagram here. Now, muscle fibers follow a principle known as the all or nothing principle, and we've talked about this in our last lecture. And basically what that means is that an individual muscle fiber either contracts maximally in response to an action potential or it does not contract at all. There are various stimuli that can act upon motor units. A threshold stimulus is a stimulus that is strong enough to create an action potential in the axon and thus an action potential in the muscle cells associated with that axon. A sub-threshold stimulus is a stimulus that is not large enough to create an action potential in the motor unit. A submaximal stimuli is a stimuli that causes increasing strength and causes reaction in more and more neurons and their associated muscle fibers. And then, as this picture shows, a maximal stimulus is a stimulus strong enough to activate all the muscle fibers in a motor unit. Muscle tone is defined as the state of partial contraction of a muscle when a muscle is not being used. And this diagram essentially shows two individuals with two different degrees of muscle tone. The person on the left obviously has less muscle tone than the person on the right. When we talk about the energy situation for muscles, we have to talk about something known as creatine phosphate. First of all, most of the energy that occurs in muscles occurs in the presence of oxygen and therefore is called aerobic respiration. This is where oxygen reacts with glucose to produce energy. Creatine phosphate is the chemical that's produced during this process in muscle, and it stores the energy in the muscles. And creatine phosphate is then used at times of energy need, the reaction proceeding from the right to the left, to produce ATP that is necessary for contraction to occur. In states of extreme stress, when you need to sprint or get away or whatever, and the time necessary to do aerobic respiration is not available, then anaerobic respiration occurs, and it produces energy, and it produces it without the need of oxygen. The downside is it's much less efficient, and it can only last for a short period of time, and then it has to stop and allow for the body to recover.